So what I'm going to be doing now is freezing down some cells that I have been growing. I need some new stocks of them. So I grew up 3 of these large plates here and 1 smaller plate just because I had extra cells. So I've already aspirated off the media, the old media on these cells, and now I'm going to wash them with this clear liquid, which is PBS, a medium-sized serological pipette, and add just enough of this PBS to cover the bottom of each plate. And the purpose of this is just to rinse away any of that old media. The next thing that I'm going to be adding is my dissociation reagent, or trypsin. It's this pink liquid here. It's in a smaller bottle, and this is basically an enzyme that's going to help the cells detach from that plate. So now that all of these are coated, they're going to go back in the cell culture incubator for 5 minutes for the enzyme to be activated. The next thing that I need to do is prep what are called cryovials. So here I have a sterile pack of cryovials. I'm not going to open this one on camera just because it is sterile. I do have one that is open already that I can show you guys. This is what a little cryovial looks like. This is basically a specialized version of a tube that allows cells or bacteria or anything to be stored at super low temperatures. So this will survive in both the -80, so that's -80 degrees Celsius, as well as the liquid nitrogen, which is at minus almost 200 degrees Celsius. They have very, very airtight lids, so none of the liquid nitrogen that they're submerged in will get into these and damage the cells or whatever is in the tube. And these are basically our vessels that we use when we are freezing cells down. We will put a little bit of cell suspension in this and then slowly bring the temperature down over a couple of days and eventually transfer it over to the liquid nitrogen. The last part of this cell freezing process is to put cells into the -80 freezer, but they actually go in a special chamber. So I have all of my cryovials that are filled with cell suspension, and that is the color, the pink is the color of the media, so they are in their original media, and then they're actually going to go in this little thing called a freezing chamber. And what this is actually filled with 100% isopropyl alcohol or 100% rubbing alcohol. And as you know, or may not know, alcohol doesn't freeze. So this basically creates a nice shield around the cells as they go through the freezing process to cool them down slowly. The cells themselves in the cell suspension will freeze, but they will freeze at a much slower pace than if we were to just throw them in the freezer. So there's a little insert where all of my cryovials are, and this goes into the freezing chamber. This is our -80 freezer. This is what's considered a coffin freezer. We do have other ones that are the upright kind. It is at -83 right now, which is good. And I will open this up. It is covered by these foam toppers. I'm going to remove one of these just to get inside. And they actually hold baskets in here where we have different boxes, and these will just have different tubes stored in them, or we even have vials of cells. I'm going to find a nice home for my cells in there. And they will stay in there for about a day or 2.